Okay. Okay. Okay, you're live with Sa Majesty Easy World. Nice to see that you're there. Today, we will take a look at JAMB biology live stream, the suspected questions. I'll take you through these 10 questions to help you and guide you in this forthcoming JAMB examination. Then you bring your questions after I've treated these 10 questions. So let's see the 10 questions. And remember to watch my live stream yesterday on chemistry, and tomorrow will also be live stream on chemistry, same time, exact place. That's Sa Majesty's World Science Channel, same time. You will still be having it. So first, take a look at the questions. Okay, you see question 1 and 2 there. Okay. Chima, don't bother. Just attempt those questions. Okay, these are the questions. Try your abilities. Okay, these are the 10 that I will handle. Then I welcome your own questions. I welcome your own questions after answering this 10, but try your ability. Let me see your answers. Let me see your answers before correction. You that is online, you see question 1. It's talking about the start codon in protein synthesis. Try if you can get an answer there. Okay, you see number 2. Number 2 is actually a calculation. Okay. This is called O-xylene. This one can now be called M-xylene, and this is called. This is the vertebral column. So we will take a look at the bones that makes up the skull, the trachea here. So that's where your ear should be. Let's Okay, welcome back. We now go to our quick note. We have the stream for today. Okay, here we'll pick those questions. We take them one after the other. I'll go with question 1. Remember, this live stream lasts only 45 minutes. By 3:15 it will be over. Okay, drop them here. Okay, see the question here. So question number 1 says the start codon in every protein synthesis. The answer is simple. The start codon in every protein synthesis is AUG. This is AUG, known as methionine. So methionine is the start codon for every protein synthesis. Remember, in protein synthesis, we have the start codon, and that is methionine, irrespective of the protein that is to be synthesized. And the base coding is adenine uracil guanine. That's adenine for A, uracil for U, and G for guanine. That's the start codon. Whereas option A, as you can see there, is actually a nonsense codon. This is nonsense, and it shows termination of a protein synthesis. Then we move straight to question number 2. So number 2 says, this is a typical calculation. In a population study of a grass species in a school farm of area 30 meters square using 2.2 meters square quadrat, a total of 1,210 plants were counted after 10 tosses. What is the population density per meter square of the grass species? So in this question, you are required just to do justice to that. You do your calculations. Let me shift this a bit and use our book. So in this case, you go back and look at what the question says. You'll be seeing it partially here. Remember, don't get confused about what is happening here. That they are just giving you some values that you might not even be using. So first, if you see this type of question, write out area of quadrat. From the question, area of quadrat is 2.2 meter square. Then next, you also now take a look at the area of the habitats. Sometimes you might not use it, but record them. That should be 30 meters square. Then after 10 tosses, which means we have the total toss is 10, and then the total plant obtained is actually 120. So for you to get the average of plants per each toss, you now say total number counted all over total tosses, which will give you 121. So you have averagely that 121 is present per 2.2 meters square, because that is the area of the quadrat. Remember that quadrat is just a square of a known dimension, something like this. This can serve as a quadrat. So in this case, the according to the question, they say 2.2 meters square, which means the area is 2.2 meters square. It means that 121 plants are found inside 2.2 meters square, but the question asks you to find the one that will be, look at it here, per meter square, not 2.2 meters square. So simple. What you have to do is you now ask yourself, if 2.2 were able to hold 121, what will 1 hold? Because per meter square means 1 meter square. So you call here x, then you cross multiply. At the end, what do we have? We have that 121 over 2.2 times 1 over 1. So your final answer is going to be 121 over 2.2. And that will give us 121 divided by 2.2. Okay. So looking at the question, let me check well. Okay, 121 divided by 2.2. We have 55. That's option D is the answer. So the correct answer is 55. This shows that we are not going to use this. We won't be using this 30 meters square that was given to you. So that one shouldn't confuse you. So if you have question or questions based on this one I've calculated, you ask. But if you don't have, I move to the next question, which is question number 3 and 4. So the next question, we are done with this. We lift our next question. It should be 4 and 3 and 4. That should be here. So look at it here. Number 3 says Darwin's finch are used to demonstrate. Simple. Darwin's finch is also called Galapagos finch. So what is the formula for that? For that what? I can't get your question. What's the formula for what? Please come again without your question. So before then, there is no formula in that population study calculation. It's all about knowing what we are required to do. So Darwin's finch is otherwise known as Galapagos finch because Charles Darwin came across these birds in Galapagos Island during his HMS Beagle. During those days, then he discovered that some of these birds have different beak modified, and they are all found in different environments within that Galapagos Island. No, that one shows exactly nothing but whereby a ground finch diversified to give rise to many species, but they are similar a bit, meaning evolution of species that are superficially different but they share common ancestor is what we call divergent evolution. And another name for divergent evolution is adaptive radiation. So looking at the option, the correct option there is option A. Adaptive radiation is the answer. Darwin's finches are used to demonstrate adaptive radiation. Remember, this provided a strong evidence to support Darwin's theory during his evolution based on natural selection, collectively with Alfred Russel Wallace. So we move over to question number 4. Question number 4 says, okay, let's shift it. Sickle cell anemia can be wiped out of existence from human population by A. Stopping existing sufferers from reproducing, B. Giving sufferers constant blood transfusion, C. Selective breeding, then D. Proper sex education. So looking at this question, simple. How do we stop sickle cell anemia? Remember that sickle cell anemia did not exist from day one. It came as a result of mutation. That is, normal hemoglobin A underwent mutation and produced this abnormal hemoglobin S, and that because that abnormality made the cells to be sickled. Actually, is a point mutation or a gene mutation, whereby in protein synthesis, remember when it comes to hemoglobin, there is what we call the globulin and the hem. And when it comes to the hem are divided into the hems are actually for one globulin molecule, that's for one hemoglobin. But the globulin have two parts. We have the globulin chain and the alpha chain and the beta chain of the globulin. So during the protein synthesis around the beta chain, I think so, there was a misconception and that led to sickle cell anemia. What am I trying to tell you? GAG and GTG caused every problem. In that case, where they're supposed to insert GAG, meaning guanine, adenine, guanine, what was inserted was GTG. So what's the difference between hemoglobin A and hemoglobin S is that hemoglobin A have this, while hemoglobin S have this, meaning just that thiamine substituted adenine, and that created sickle cell anemia. So we can wipe them out of existence by making use of the knowledge of selective breeding. Selective breeding is that thing they say marriage counseling. You should know your genotype before marrying, so that the possibility of having these people coming out again will no longer be there, so that eventually when those that are suffering eventually dies, the human population will be free from sickle cell anemia. So the correct answer is by selective what? Breeding. Proper sex education to sufferers will not help out. Giving sufferers constant blood transfusion, that will manage it, not to take it out of existence. So that is it. If you have questions or questions you ask, I'm moving to the next question. Okay, but before that, remember what I said, that sickle cell anemia can be wiped out by selective breeding, meaning that is what we call counseling and doing your marriage test before the marriage. You don't just go and continue doing your go on with your marriage, meaning AS should not marry AS, and by doing so there won't be possibility of having these sicklers again in our population. So it is possible to be wiped out of existence. Now we take a look at the next question. Okay, before then, let's remove this. Okay. So that's question 4. Okay, we look at 5 and 6. Now, question 5. Which of the following is not true about mammals? Remember, this might be confusing a bit. Mammals are ectotherms. B. Some mammals do not have placenta. C. Some mammals can fly. D. Some mammals lay egg. So if you look at these options, someone might choose wrongly. Check out option A means that mammals are cold-blooded, and this is not true. Mammals are not cold-blooded. So among these options for question number 5, option A is not correct about mammal. Of course, some mammals don't have placenta, and those mammals without placenta, they exist, of course, and they are the ones we call the marsupials. We have the marsupials, that's the marsupial mammals like the kangaroo. They don't have placenta. Then option C says some mammals can fly. Of course, some mammals can fly. The Chiroptera, like the bats, can fly. Then some mammals lay egg. Yes, we have egg-laying mammals known as monotremes. Good examples are the duck-billed platypus and spiny artiata. We move to question number 6, which says, which of the following bones lacks body? This is quite interesting. The bones that lacks body. When we say body here, according to maybe if you are in O level, you should see that this body here means lacking centrum. So the bone that lacks body is actually atlas. Atlas don't have anything like body. So the correct answer is this. So atlas lacks a centrum. Then I'll show you something here so that you understand what we mean by atlas. I'll use 3D anatomy to illustrate this I've just said now. Where do we have it? Okay, this 3D anatomy. Here I'm going to isolate the atlas. I'll remove. Okay. All the connective tissues removed. That's an atlas. I have to isolate it. So this is a typical atlas. There is nothing like centrum. This is where the body is supposed to be. And what protrudes into this space is what we call the odontoid process of the axis, or what we call the dens of the axis. So atlas lacks body in human being because actually the part that's supposed to form the body is giving space so that the odontoid or the dens of the axis can do what? Articulate. So this is a typical atlas. I'm going to highlight another. Okay, the second cervical bone is actually the axis, as you can see here. I've highlighted the axis. So in isolation, you see there is a body, there is thickness, there is a thick base. You see, it has a body, unlike atlas. So among the bones listed in that question, it is the atlas that lacks centrum. That is, atlas lacks a body. We now go back to our note. So question number 6 answered. Okay. Remember that you're still live with Her Majesty. You can bring your questions when necessary. Then, you should endeavor to come and join us in everyday live stream. Every time it is on your clock, if you reach 2:30 p.m., that's GMT plus 1 Nigerian time, you come and join me here on chemistry and biology. Tomorrow, same time, I will take you on JAMB chemistry. Remember to come with your questions. So then, before we proceed, as you can see here, you can get your word registered with this school. It's a nice school, Fountain Comprehensive College, Enugu, raising a total child. You see where they are located, you find them. I've worked with them. It's a very nice school, of course. So get your child registered. They raise your child, of course. So thank you, Fountain Comprehensive, for partnering with Her Majesty. So we go back to what we're doing. So far, so good. The questions we have answered. We say that the answer to this question is B. That's number 1 is B. Number 2 is B, 55. The number 3 is A, adaptive radiation. The number 4 is selective breeding. The number 5 we said is A, mammals are ectotherms. Then here we need to lay more emphasis. Question number 7 says osmosis is best defined as A, the movement of water molecules from a region of higher concentration to a region of higher concentration of water molecules to a region of lower concentration of water molecules through a semi-permeable membrane. B says movement of water molecules. Just watch the options here. This is where students are taught wrong thing. If you are not careful here, you see what comes into your mind. When they say osmosis and diffusion, you say that osmosis involves moving from low to high, while diffusion involves moving from high to low. Please, that is a virus and is a wrong way to teach you. By so doing, I'm sure you're going to choose option B as the correct one. Now, osmosis is the movement of water molecule from region of lower concentration to region of higher concentration through a semi-permeable membrane, but unfortunately that is a wrong definition of what? Osmosis. Please, option A is the correct definition of osmosis. Please, osmosis is a passive transport. So I'll go back to my note and show you what I mean by passive transport. Okay, we shift it aside. Now, what we are trying to say is water molecule is what is moving. Let me use this to show a setup for osmosis. If you have something like this, then you place your yam inside. Here is your distilled water. Then let's take that the inside is now sugar solution or salt solution. So in labeling, here is this is the peeled yam or potato, and that is semi-permeable. Then here is the distilled water. Then here is our sugar solution or salt solution. So to make it clearer, let's have two partitions here. This is region A, and we take here to be region B. Let me use a separate color. Okay, we take here as region A. Here is our region B. So watch this, A and B. Region B shows only water. Only water molecules are there. And by measuring the concentration, it shows that water molecule concentration here is 100%. But here is mixture of sugar and water. So Region A is sugar solution, meaning sugar and water mixed together. So when you look at this, it shows that water and sugar are mixed up. So the percentage of water here can never be 100 because the water and sugar are sharing it equally. Therefore, as you all know, in this region B, concentration of water is very high. It's 100%. That means there is nothing else here apart from water. So concentration of water here is high, while concentration of water here is low. But solute concentration in A is high. Watch it. This one highlighted in yellow is for concentration of solvent. But when we have another condition for solute, let's say sugar here, the concentration of sugar here is high and the concentration of sugar here is low. Why? Because there is no sugar here at all. So in this case, the purple highlight is for sugar concentration, while the yellow, the one highlighted in yellow means water concentration. Remember, I'm describing your osmosis setup. Now, in this case, we all know that water will move from the bath or the bowl into the yam cavity or potato across the peeled part of the yam. So in this case, it is quite clear that the movement of water is going to be this way. Water will move from B to A. And if you want to describe this movement, why will you say that osmosis is the movement of water molecule from region of lower concentration? When you say water molecule, the only molecule there is water. That's the only thing you have mentioned, movement of water molecules from region of lower concentration to region of higher concentration. Please, this definition means that water will move from where it is smaller to where it is high, which is not true in real life. So water will definitely move from A to B. So for you to define this, you can say that osmosis is the movement of water molecule from the region of higher concentration of water to the region of lower concentration of water through a semi-permeable membrane. So here is standing as a semi-permeable membrane. The line here is a semi-permeable membrane. Or you can say osmosis is the movement of water molecule from the region of lower concentration of solute to the region of higher concentration of solute across semi-permeable membrane. But don't define osmosis as the movement of water molecule from region of lower concentration to region of higher concentration. You need to be specific. You can define osmosis high to low or low to high. So osmosis is a passive transport. Therefore, it depends on concentration gradient, meaning molecules move from where they are high to where they are low, and that's what we call passive what? Transport. So please, looking at that question, the correct answer is A, the movement of water molecule from region of higher concentration of water to region of lower concentration of water through a semi-permeable membrane, which means you can confidently define osmosis as the diffusion of water molecules. So osmosis can be seen as diffusion of water molecule. Don't get it confused. If you have more questions, see you there in the comment box, or I'm here for you for your live chat. We now go to the next question. The next question should be question number 8 and 9. So that's it here. So this question, which of the heart chambers contrasts to supply pulmonary circulation? The answer is, when we say pulmonary circulation, is actually movement of blood or circulation of blood from the heart to the lungs and then from the lungs back to the heart. So the particular chamber of the heart that is responsible for pulmonary circulation supply. Remember that ventricles collectively supply blood outside the heart. So never you answer auricles when the supply of blood is outside the heart. So the chambers that pumps to supply blood outside the heart are called ventricles, but auricles only supply within the heart. So with regard to this, we are looking at either option B or option D. Option B says left ventricle, while option D says right ventricle. Now, it is good for you to know that the thickest chamber of the heart is actually the left ventricle, and it supplies blood to longer distance. So it doesn't supply to the lungs, it supplies to other parts of our body apart from the lungs, which means our answer becomes automatically B. So left ventricles contract to supply the pulmonary circulation, and pulmonary circulation is the circulation from the heart to the lungs, then from the lungs back to the heart for what? oxygenation. That's the significance of pulmonary circulation. If you don't call it pulmonary, call it you can call it lung circulation. And the significance is to enrich the blood, to nourish the blood with fresh air. In that case, the blood becomes more oxygenated. Then question number 9 say the enzyme that catalyzes the first phosphorylation during glycolysis. Remember, glycolysis is the conversion of glucose to pyruvic acid, and there is a 10-step reaction divided into preparatory and energy reading stage. Watch glycolysis here in my channel. I have the details. Go under the playlist biochemistry, you see it. The answer to this question is hexokinase. Hexokinase, that's option C for question number 9 is the correct answer. We move on to the next question. That's question number 10, and we wrap it up for today's live stream. Okay. Question number 10 says, the spermatophytes are divided into monocots and dicots, B, gymnosperms and angiosperms, C, vascular and non-vascular plants, D, conifers and ginkgos. The correct answer is B, gymnosperms and angiosperms. Spermatophytes means seed plants. In Greek, sperma or sperm means seed and phyte means plant. So spermatophyte just means seed plants. So the correct answer here is gymnosperms and angiosperms. So seed plants are divided into non-flowering plants, which are gymnosperms or gymnosperms, then flowering plants, which are also called angiosperms. Don't be tempted answering monocots and dicots. Monocotyledons and dicotyledons are collectively angiosperms. They are all flowering plants. So that wraps it up for today's live stream. It's nice having you around today, and don't forget to also queue up tomorrow for your chemistry live stream, and bring your questions alongside as you are coming. The Her Majesty's World Science channel is here for you, and you are going nowhere better than this particular channel. So we expect that you should do more, and remember to share my videos also.
We're here today to ask you to please subscribe to Sir Majesty Easy World. It has cool experiments and it makes science fun. So, subscribe. Please. I love you. Thank you for watching.